wavelength photons, basically, or light particles, how it is they're being reflected or diffracted off of some molecule. And what she found was a picture like this. So Watson and Crick got that picture and they thought, well, really what that looks like is the DNA molecule forms a twisted helix, right? A double helix. There are complementary strands that are spinning in space. And so from that X-ray crystallograph, Watson and Crick were able to accurately describe what the molecule looks like. Both of them were, of course, awarded the Nobel Prize. So when I was applying for PhD programs, I got accepted to the University of California, San Diego, which is where James Watson was working at the time. So we got a chance to meet this guy, and he's really pompous. Not somebody I would have wanted to work with. But I suppose if I won the Nobel Prize, I'd be a little bit pompous too. So
How old is he?
He's old.
He's still alive?
No, I think he passed recently, but
Oh, okay.
I started graduate school, well, PhD back in 2006. So he was alive at that point. Okay, so what we know, so these are the periods of pyrimidines, right? So we have purines, A's and G's, and pyrimidines are T's and C's. And we know that A's, C's, G's, and C's are the DNA nucleotides. With RNA, it's a little bit different. Instead of T's, we have U's. But that U is still considered a pyrimidine. It looks similar. There are just some slightly different conformations within this particular ring. Okay, so if we were to kind of create like a line picture or a line diagram of the DNA molecule in a two-dimensional space, it would look something like this, where there are alternating phosphates and sugars, right? Because that kind of constitutes the backbone. And when we have those phosphodiester bonds that are created between nucleotides, they're really created between a phosphate, a covalent bond between a phosphate and the sugar. But what's interesting is they kind of alternate like this. And it gives the DNA molecule some sidedness. So we have phosphate, then sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, like that. But here, where this strand started with the phosphate, this complementary strand starts with the sugar. And you get converse alternating pattern, like that. And then coming off in between phosphates and sugars, we have our A's, T's, G's, and C's, these nitrogenous bases. And it's going to be hydrogen bonds between these that hold those complementary strands together. Okay, so let me add one more layer of complexity to this. Depending on which molecules at the top, either the phosphate or the sugar, will determine its side. So, I don't want to go there yet. So there is what's called a 5' and a 3' side to the DNA. If the sugar is on the top, I believe this is called the 3' side. And it goes from 3' down to 5', because there's a phosphate at the bottom. And we know on this end, because there is a sugar, it's got to be a 3' and 5' like that. Now, the reason that that's important is because our cellular machinery can only read in one direction. It only reads from, I think, 5 to 3'. And I'll make sure that these facts are true in just a few minutes. So what that means is, if there's some particular gene sequence right here, then our cellular machinery has to read from 5' direction down towards the 3', like that. And if the gene region is over here, then it's got to read in the opposite direction. And that becomes particularly true when we're replicating our DNA during the S phase of the cell cycle. Okay, so let's press on a little bit. Now, are there questions so far? Yeah, Kira.
So what is the red and the blue? I'm sorry.
So the red is a phosphate,
Okay.
and the blue is sugar. Yeah, I tried to make a correspondence to that little nucleotide that I've drawn over there on the other side of the board. Okay, now I need you guys to really remember this point. That is that DNA exists as complementary strands, not as mirror images. Mirror images and complementary strands are quite different, right? So my hands exist as mirror images of each other. If I was to put my left hand up to the mirror, it would look like my right hand, right? And that's really determined by the orientation of my thumb. That's very different than the way that DNA molecules exist. So, for example, if I had some non-random sequence of DNA and it looked like this, like that, a mirror image of that would just be A, T, T, C, oh, C, C, G, T, A, right? It would just be this sequence backwards. But that's not how DNA works. It exists as a complementary strand. And so instead of a mirror image, it's a complementary strand, which means that this is going to be T, A, G, C, G, G, A, T. That sequence and that sequence are very, very different. But the question is, who cares? Why is that an important concept? It's an important concept, you guys, because that effectively doubles the amount of genetic material available to us. Remember, DNA can only be read in one direction. And if it is from 5' to 3', that means that this sequence is going to code for the same proteins as this sequence is, just read in opposite. However, if we have complementary strands, this sequence of DNA codes for a very different protein than this sequence of DNA. So the real novelty, the real benefit of having complementary strands is being able to increase the amount of genes within a genome. That's an important concept.
Can you say that one more time? Mr. Shaw?
Sure. The important thing about having complementary strands is that effectively you double the amount of genes within a genome. Significantly increase it. Your textbooks refer to it as mirror images, and it is just blatantly false. Please don't make that mistake. Okay, so this is how our DNA looks, right? It exists as complementary strands, and the question is, how do we replicate our DNA? And there are three models that I'm going to want you to be familiar with within DNA replication. The first of which is called a conservative model. In conservative model, what that means is you're going to start off with a double helix, and at the end, you're going to have that exact same double helix, so that double helix is conserved, and a daughter double helix that consists of entirely new daughter DNA. Now, I know these diagrams are drawn on the board, but just for the sake of example, I want to draw them myself. And is it okay? Well, I'll erase this side over here. Is it okay if I erase this? We're going to fix that. Okay, so if this represents parental DNA, and during the S phase we know we want to replicate that parental DNA, then what we're saying happens is each of these strands of DNA acts as a template. So they're going to separate like this. And allow our cellular machinery to make a complementary strand on the other side of it. So that at the end, it's going to look like this. And this red strand represents some new daughter DNA. And then because it's the conservative model, what's going to happen is these two strands of DNA are going to separate again, and this one is going to reattach to this one, and this one is going to attach to that one. So that at the end, we have our parental DNA back, and then we have an entirely new strand of daughter DNA over here. I should draw these arrows a little bit differently so that makes more sense. So this one comes over here, this one comes over here to make that. This one comes down here, and this one comes over here, like that. This is the conservative model. So that's option number one, and how it is that we create new strands of DNA. Alternatively, we could have the semi-conservative model. Next, it looks very much like the conservative model, where we start off with some parental DNA. And then we get separation of complementary strands so that there is a template for new DNA. Like so. And then we get new daughter DNA that forms the complementary strands to each of those template pieces. And that's the end of it. So that each daughter strand of DNA consists of a parental strand and a complementary daughter strand. And this is the semi-conservative. And then the last model suggests that each daughter strand of DNA, each complementary strand within those daughter strands of DNA is going to be a mosaic of both parental and daughter DNA. So we're going to get some kind of segregation of DNA, and it's going to consist of random regions of the DNA molecule, like this. And the same thing over here. So that each of the resultant DNA molecules is a mosaic of parental and daughter DNA. And this is the dispersive model. Suggesting that parental DNA is dispersed across the daughter DNA strands. Oh, I should go.
Is this a random assortment?
Yeah, you can think of it as a random assortment. Okay. So these are three possible models whereby DNA can be replicated. If you had to choose, which one of those models do you think looks most correct?
Conservative.
The conservative?
I think
Or maybe the dispersive.
Dispersive? Any
Yeah.
others? It's actually the semi-conservative.
Oh, wow.
Yeah. And Again, what we need to do is apply the principle of parsimony, or the simplest, Occam's razor. The simplest explanation is generally the correct one. I mean, the simplest way to create new daughter strands of DNA is to use parental strands as templates, right? It's just separate them, replicate them, separate them, replicate them. And here what we're doing is we're separating and replicating, and then we're separating again and reattaching, which is a little bit more complex than this one. And here we're going to have to use restriction enzymes that come in and chop up DNA. And you can imagine making sure that all of the DNA consists, or is represented across the entirety of these DNA molecules would be a pretty onerous task, right? Yeah.
So during all of these different models, is the parent strand actually breaking apart to create a template, or is it just a copy of it?
So within the semi-conservative model and within the conservative model, these two strands are actually separating from one another. So the hydrogen bonds between complementary nucleotides are actually being physically broken. That'll also happen in the dispersive model. And remember, these were all theoretical. These are just possible ways in which this could occur. That would also happen within the dispersive model, but in addition to that, you're also having restriction enzymes that are coming and breaking bonds between phosphates and sugars. Okay, so on an exam, would you guys be able to tell me at least two of the models, if not all three of the models of DNA replication, and then tell me which one of them is correct? Like you'd actually be able to say, okay, so within the conservative model, this is what it looks like and this is kind of what's happening. And within the semi-conservative model, this is what's happening and this is kind of what it looks like. And we think the semi-conservative model is correct. Okay, so be ready for an item like that as a written item. Okay, so the semi-conservative model is correct. And what we need to do is talk a little bit about how it is DNA is actually replicated using the semi-conservative model. Okay, so did I do it correctly? I did. So remember, there is a 3' and a 5' side to the DNA molecule. If the end of